Plant cells are organized in the same way as the cells of other multicellular organisms. Cells are grouped into tissues, tissues are grouped into organs, organs are grouped into organ systems, and organ systems work together to keep the organism alive. Plants are made of organs like stems, roots, and leaves. Plant organs work together to make organ systems. These can perform the various tasks that a plant needs to carry out to survive and grow. For example, transporting substances around the plant. Plant organs are made of tissues. Here are some examples. Epidermal tissue. This covers the whole plant. Palisade mesophyll tissue. This is the part of the leaf where most photosynthesis happens. Underneath is the spongy mesophyll tissue. This is also in the leaf and contains big air spaces to allow gases to diffuse in and out of the cells. Xylem and phloem transport things like water, mineral ions, and food around the plant, through the roots, stems, and leaves. Meristem tissue is found at the growing tips of shoots and roots, and is able to differentiate, change, into lots of different types of plant cells, allowing the plant to grow. The leaf is an organ made up of several types of tissue. Leaves contain epidermal, mesophyll, xylem, and phloem tissues. You need to know how the structures of the tissues that make up the leaf are related to their functions. The epidermal tissues are covered with a waxy cuticle, which helps to reduce water loss by evaporation. The upper epidermis is transparent so that light can pass through it to the palisade layer. The palisade layer has lots of chloroplasts, the little structures where photosynthesis takes place. This means that they're near the top of the leaf where they can get the most light. The xylem and phloem form a network of vascular bundles, which deliver water and other nutrients to the entire leaf and take away the glucose produced by photosynthesis. They also help support the structure. The tissues of the leaves are also adapted for efficient gas exchange. The lower epidermis is full of little holes called stomata, which let carbon dioxide diffuse directly into the leaf. The opening and closing of stomata is controlled by guard cells in response to environmental conditions. The air spaces in the spongy mesophyll tissue increase the rate of diffusion of gases. Phloem tubes are made of columns of elongated living cells with small pores in the end walls to allow cell sap to flow through. Cell sap is a liquid that is made up of the substances being transported and water. Phloem tubes transport food substances, mainly dissolved sugars made in the leaves, to the rest of the plant for immediate use or for storage. The transport goes in both directions, from the leaves down to the roots and from the roots up to the leaves. This process is called translocation. Xylem tubes are made of dead cells, joined end to end with no end walls between them, and a hole down the middle. They're strengthened with a material called lignin. Xylem tubes carry water and mineral ions up the plant, from the roots to the stem and leaves. The movement of water from the roots through the xylem and out of the leaves is called the transpiration stream. Transpiration is the loss of water from a plant. The transpiration stream is the movement of water through a plant from the roots to the leaves, and it happens like this. Water from inside a leaf evaporates and diffuses out of the leaf, mainly through the stomata, the tiny holes found mainly on the lower surface of the leaf. This creates a slight shortage of water in the leaf, and so more water is drawn up from the rest of the plant through the xylem vessels to replace it. This in turn means that more water is drawn up from the roots, and so there is a constant transpiration stream of water through the plant. Transpiration is just a side effect of the way leaves are adapted for photosynthesis. They have to have stomata in them so that gases can be exchanged easily. Because there is more water inside the plant than the air outside, the water escapes from the leaves through the stomata by diffusion. Transpiration rate is affected by four main things. Light intensity. The brighter the light, the greater the transpiration rate. Stomata begin to close as it gets darker. Photosynthesis can't happen in the dark, so they don't need to be open to let carbon dioxide in. When the stomata are closed, very little water can escape. Temperature. The warmer it is, the faster transpiration happens. When it's warm, the water particles have more energy to evaporate and diffuse out of the stomata. Airflow. The better the airflow around the leaf, for example stronger wind, the greater the transpiration rate. If airflow around the leaf is poor, the water vapor just surrounds the leaf and doesn't move away. This means there's a high concentration of water particles outside of the leaf as well as inside, so diffusion doesn't happen as quickly. If there's good airflow, the water vapor is swept away, maintaining a low concentration of water in the air outside the leaf. Diffusion then happens quickly from an area of higher concentration to an area of lower concentration. Humidity. The drier the air around the leaf, the faster transpiration happens. This is like what happens with airflow. If the air is humid, there is a lot of water in it already, so there is not much of a difference between the inside and outside of the leaf. Diffusion happens fastest if there is a really high concentration in one place and a really low concentration in another. You can estimate the rate of transpiration by measuring the uptake of water by a plant. This is because you can assume that water uptake by the plant is directly related to water lost by the leaves. This diagram shows the apparatus that you will need. It shows a potometer. This is the apparatus that we use for measuring the uptake of water by a plant. In your exam, you might need to read off data related to transpiration from a graph, chart, or table and explain what the data is showing. You also need to be able to create your own graphs with suitable scales and axes. To investigate the rate of transpiration, set up the apparatus as in the diagram, and then record the starting position of the air bubble. Start a stopwatch and record the distance moved by the bubble per unit of time, for example per hour. Keep the conditions constant throughout the experiment. Keep the temperature and the air humidity, for example, the same. Calculating the rate of transpiration is easy once you have the data. You can use the data you collect in your experiment and use this equation: rate equals distance moved divided by time taken. 2 guard cells surround each stoma. They have a kidney shape which opens and closes the stomata in a leaf. When the plant has a lot of water, the guard cells fill with it and go plump and turgid. This makes the stomata open so the gases can be exchanged for photosynthesis. When the plant is short of water, the guard cells lose water and become flaccid, making the stomata close. This helps stop too much water vapor escaping. Thin outer walls and thickened inner walls make the opening and closing work. They're also sensitive to light and close at night to save water without losing out on photosynthesis. You usually find more stomata on the underside of the leaves than on the top. The lower surface is shaded and cooler, so less water is lost through the stomata than if they were on the upper surface. Guard cells are therefore adapted for gas exchange and controlling water loss within a leaf. You can use a light microscope to investigate the distribution of stomata and guard cells on a leaf. For some plants, you can peel the epidermal tissue straight off the leaf and mount it on a piece of microscope slide. Using a pipette, put a large drop of water onto the slide and then place the tissue into the water using tweezers. Remember to put the cover slip on top. For a leaf where peeling the top layer off isn't possible, you need to make an imprint on its surface, and you can do this using clear nail varnish. Apply a thin layer of varnish to the underside of the leaf and allow it to dry. Then use a piece of sticky tape to remove the varnish from the leaf by gently peeling it off, and stick the sticky tape with the attached nail varnish imprint onto your microscope slide. Now you can view your slide. Remember how to use a microscope. Put the slide on the stage and focus the microscope. Start with the least powerful objective lens and work your way up until you can see the stomata and the guard cells clearly. You could draw a labeled diagram of what you can see down the microscope. You can estimate the total number of stomata on the surface you're investigating. It would take ages to count every stoma on the surface, so it makes sense to just count a sample. Count the stomata that you can see in your field of vision, the bit of the specimen that you can see when you look down the microscope. When you've finished, move the slide so you're looking at a new field of vision and count the stomata again. Do this at least 3 times and calculate the mean to determine an average number of stomata per field of vision. To estimate the total number of stomata on the surface that you're investigating, you need to work out how many times the field of vision would fit into it. Find out the area of the field of vision and divide the total area of the surface by it. Finally, multiply your answer by the average number of stomata per field of vision. This gives you an estimate of the total number of stomata on the surface. You can easily compare the distribution of stomata in different areas of the leaf, for example the upper and lower surfaces, or between different leaves. To do this, carry out the same steps for each of the leaf surfaces you're investigating and compare that mean number of stomata per field of vision on each of the surfaces. This video was twice as long as normal, but does cover all of the information about plants that you need to know for Module 2. So remember to go back onto Google Classroom, answer the question that comes along with this video. This is the end of the topic, so remember that there will be a test in class coming up, and you need to know all of this information about plants for the topic.